So I'm the other co-chair of the Task Force 12 that talked about compositional analysis. So it's important to remember that there are a number of different studies that go into the safety assessment of GM crops. Compositional analysis itself is not the safety assessment, but information that's gathered during a compositional analysis study is used in the overall safety assessment, along with information from the other studies, to evaluate whether a novel food or feed resulting from a GM crop is safe or not. So as Angela mentioned, it's recognized that unintended changes result from conventional breeding methodology. However, the majority of regulatory bodies consider conventional non-GM varieties to be safe for consumption because it's based on a history of safe use in terms of the context of traditional preparation methods and the amount of a crop that is consumed in the diet of an animal or a human. Compositional analysis studies are used to assess the levels of different nutrients, anti-nutrients, toxicants, any other appropriate secondary metabolites that may be in the resultant food and feed, and put that in relation to the natural variability within the conventional crop itself. Compositional analysis studies are used to document the types of possible changes that may occur, and these are often classified as intended or unintended. Intended changes, that's pretty obvious. Those are the changes that are really the goal of inserting the genes, such as higher oil content, insect resistance, herbicide tolerance, etc. Unintended changes may also occur. It's hypothesized that these might occur due to the insertion of DNA. However, as far as I know, that really hasn't occurred. Or products of the transgene or transgenes could act somewhere else in the plant and cause changes. And so compositional analysis helps us to identify these. We can also break down the unintended effects into expected and unexpected. So expected would be ones that we could hypothesize might occur. So let's say we're modifying a particular fatty acid level. We're trying to raise a fatty acid level. You could hypothesize that other fatty acids would change too. So these would be unintended effects, but we would expect that they were there based on hypothesis. There also could be unexpected changes. In these, it's difficult to come up with a hypothesis for. So you might modify a particular pathway, but yet some vitamin changes that's unrelated and you don't know why. So that would be an unexpected, unintended effect that compositional analysis studies can document. So in compositional analysis studies, we can compare levels of components in the GM crop to conventional levels in the conventional crop with the idea to demonstrate substantial equivalence. And it's important to remember that substantial equivalence does not mean that they're identical. It just means that you could replace one with the other, variety 1 with variety 2, soybean or corn or whatever it happens to be, and you really would not notice any difference in the dietary intake. So just to illustrate what we might be looking at, here are two different varieties of soybean, and you see two different graphs. One is protein content and the other is carbohydrate content. The green bars are where the mean lies. So you see in protein content there's some difference in the mean, and the same with in the carbohydrate content. And then the long lines represent the ranges of values. And these were over 8 locations with 4 reps at each location. So in protein, you can see, the mean's a little bit different. The magnitude of the range is similar. However, there is a little bit of difference in the range. In carbohydrates, you see variety 1, the magnitude of the range is much greater than in variety 2. However, you could substitute variety 1 for variety 2 in terms of dietary intake, and you really wouldn't see any safety issues or any change in the diet. So you would say that these two varieties, although they're different, they're substantially equivalent. So what do we measure during crop composition studies, and how do we decide what we need to measure? Basically, we usually consult the Organization for Economic and Cooperative Development consensus documents. So they have this OECD has come out with a document for the particular crops that we're working with. So obviously the major ones of maize, corn, soybean, but also some not so major. And what they've done in these documents is they've assessed the levels of key nutrients, anti-nutrients, and toxicants, and they put this in context with the contributions that crop makes into the diet. So they point out which ones probably should be surveyed to see if there may be an impact on the nutrition of the crop. Also, it depends on the nature of the traits of interest themselves. So if you're changing a particular pathway, you probably want to look at some other components within that pathway to see what changes may be there. But really, coming from the workshop, it was understood that the decision on what to include really should be hypothesis-driven and not just a shotgun approach of anything and everything. You really should think about what the trait or traits are doing and assess what's meaningful for that particular trait. So just to work through a compositional analysis study in order to demonstrate substantial equivalence, we would grow the non-GM crop along with a comparator. Traditionally, this is a near isogenic, non-transgenic variety. And we measure the different components of interest, and then we compare those statistically. If there are no differences, then we can conclude that on a nutritional basis, the crop is as safe as the conventional crop, and then that conclusion feeds into the overall safety assessment. If there are statistical differences, then we may need to investigate further what those differences mean. And often the way we do this is by comparing the variability within the comparator itself, within the GM crop, the conventional crop varieties that may be grown in the same trial. Also, we'll compare with literature values or values in databases. And I'll talk a little bit more about the values in the databases and how important they are for capturing the variability within the crop. The conclusions from these comparisons then again feed into the overall safety assessment. So the databases themselves have become very important tools in helping to describe the boundaries of natural variability for each crop. Because if you remember, the levels in the conventional crop are considered to be safe. So we want a way to capture all the variability within a particular crop. The nice thing about the databases is that they're easily accessed and they're updatable. So values that may be recorded in the literature are a little bit static in that they're grown in particular environments. You may have had 5 locations, 6 different varieties, et cetera, but still that's all you have, whereas the crop itself is grown over multiple years, multiple locations, multiple geographies, environments, agricultural practices, and that all adds to the variability, as Angela pointed out. So it's very important to have these compilations of data in these databases to help assess that variability. So just as an example of what can happen, Seguin et al. Working in Canada showed that mid to late May seeding of soybean across 4 different locations resulted in a 45% greater alpha-tocopherol or vitamin E levels than seeding at later dates. So just a planting date is going to make a big difference. So when we conduct studies, we tend to plant everything the same. However, if that crop were delayed in planting for some reason, it would change. Databases are able to pull all those data together and show all the variability that's possible for the crop. So Alba et al. Pulled data from the ILSI database, and the nice thing about this is they binned the data and came up with histograms that show what the expected variability would be. So if you look at total fat, you would expect less variability in fat compared to something like crude protein. You see a wider variability within that particular crop. So that when we're assessing the safety of the crops, we're a little more tolerant of changes in crude protein compared to total fat. So the chances that we have extreme levels in crude protein are actually natural for the crop. So just to mention some of the databases, of course, our ILSI database at cropcomposition.org. It's very user-friendly. In Japan, they've also come up with a database looking at rice and soybean seed. And actually, they've included not only comprehensive data from field trials that were conducted within Japan, but also they recognize that really these databases need to reflect what's present in the marketplace, not just what's present in a particular experiment. So they actually purchased rice on the market in Japan, soybeans are imported into Japan, so they included not only Japan, but also some samples from U.S. grain, and Brazil and China as well. And the importance of these databases are recognized for people working with other crops, not just the big crops, maize and soybean and cotton. But in Malaysia, they've started developing an oil palm database because they realize that they need to be able to capture the variability within the crop. Embrapa has worked in Brazil in bean. They have their data together. And also in India, they talked about putting together an eggplant database. So these data are very useful in these databases. I think I skipped. Yeah. So one of the questions that we tackled during the workshop was, what is the appropriate comparator to use in these studies? Traditionally, as I mentioned earlier, this has been a non-transgenic, near-isogenic comparator. However, near-isogenic doesn't mean necessarily that it's identical. So right off the bat, you may have some changes, subtle changes within the control in your experiment and the transgenic that you're making the comparisons with. So if the goal of the new trait is to purposely alter the composition, such as if we just use the example of modifying a particular fatty acid, maybe we're upping oleic acid or something, the profile no longer is the same as the crop. So the best comparator may actually be, for a safety assessment, may be a completely different crop. And it was recognized in the workshop, as we start working with complex stacks of traits and how those are integrated, the appropriate non-GM comparator may actually not be available for these direct comparisons. So these reference varieties are commercial, conventional varieties that have a history of safe use. However, for some major crops, the GM crop varieties constitute 90% or greater of the varieties grown in a particular region. And also, commercial conventional varieties may not be readily available. A lot of companies are no longer producing the non-GM to be marketed. They produce a non-GM variety, and then they integrate their top traits within that. So you've got the best germplasm and the best traits. And so what's happening is in agriculture, especially in certain countries, you really don't have the non-GM varieties available. So now what do you compare to? You have these old varieties, which may have different variability or different amounts of nutrients, and then you have these new varieties that could be completely different. So you're really not comparing the same thing. So conventional non-GM varieties with a history of safe use may no longer be representative of the commodity that's being sold on the market. And that's really what we're trying to do, is assess our traits in terms of what is in the market. So we have a greater than 10-year history of consumption of GM crops without any recorded adverse effects. So one of the questions that came out of the workshop was, when are these GM crops going to be considered to have a history of safe use? Because if we could use those in our studies, then that would actually reflect what's in the actual marketplace, what's being grown by the growers and being marketed around the world. So similar to what Angela had as conclusions, we've had 15 years of experience working with safety in GM crops, and perhaps compositional analysis approach could take a different focus depending on what we have learned. Do we need data in all cases? So we have a lot of history with maize, also with soybean. Perhaps these crops don't need to be as heavily scrutinized as a new crop. When we start working with a new crop, we don't have the experience, maybe we need to do the full compositional analysis. Also the traits, we have a lot of experience with glyphosate-related herbicide resistance, with BT genes, and we've been able to show that those are safe, they're not making the changes. So perhaps when we're dealing with those types of traits, we don't need to scrutinize those as heavily as, say, a completely novel trait, which may need more scrutiny until we learn more about it. We also might want to modify the OECD suggested list or provide input to this group, depending on the trait and experience with the crop, so that the decisions on what we are measuring are hypothesis-driven. So whereas regulations have remained relatively static, the science in the seed industry really has progressed. And the general feeling coming out of the workshop was that what we know from the science really should reshape the regulatory environment going forward. And so with that, I'll take any questions that we happen to have.